Good afternoon. Welcome back to Zoology second year class. Before we are in SAQs, so the immune system SAQs we are having. Last question is left. Last SAQ is left. That is HIV and AIDS. Before that, we discuss about the B cells. We discuss about the already we discussed about B cells. We completed, and the next the immunoglobulins. Second SAQ, immunoglobulins. Next, third one is the humoral immunity. Cytotoxic or cellular immunity. Cytotoxic or the cellular immunity. Here, these four we already discussed. Next, last one is the in the immune. Cytotoxic immunity. Next, last one is the HIV. HIV, the AIDS. Here. Now the last one is left that we are going to discuss. Here, see here. All of you know this one about the AIDS and the HIV. The virus, human immunodeficiency virus, causing the mainly the AIDS they are causing. That is called as a acquired immunodeficiency syndrome. Clear? Next, what it is means in this the HIV means the human immunodeficiency virus. Here, this is the full form of the HIV is the human immunodeficiency virus. It is transmitted, which how it is transmitted means the sexual contact and the blood transfusion means blood is transferring from one person to another person, and the sharing the needles means the sometimes injections are the sharing the injections, the needles sharing time also they affected needles of the infected persons and from the infected mother to child means the blood transfusion means mixing of the blood or the fluids that time only that is transmitted. Here it is transmitted by the sexual contact, blood transfusion, sharing of the needles of the infected person, and from the infected mother to child it is infected. Here what about the mechanism means the see here the after the entering of the HIV is entering inside of the body, the TH cells and the macrophages and dendritic cells are identify this one. First, when the HIV is enter in human body, immediately T helper cells, these are the one type of the T cells, T helper, T cytokinin cells are there, like that only. The T cells, one type of the cells, they are called the TH cells, and the macrophages and the dendritic cells are identifying that HIV first. Clear? After that, what will happen means the HIV virus, the structure I will show the structure. Wait. See here, this one is the actual structure of the HIV virus. See this one, the center, the ssRNA is present. The genetic material is present. That genetic material is called as a ssRNA is present. That is surrounded by the ssRNA is present. That is surrounded by the, see this one, this is the center. This is surrounded by the lipid, the first lipid layer is present. Surrounding of that, the lipid layer is present, and the first protein coat, sorry. The first, the rRNA is surrounded by the protein coat. Protein coat is surrounded by the lipid coat. Here, this is the genetic material, ssRNA. This dark structure is in this is called as a reverse transcriptase enzyme. Reverse transcriptase, reverse transcriptase enzyme, and this is covered by the cell that the coat that is called as a protein coat is there. After that, that is surrounded by the lipid coat is there. Inside of that lipids are they are having the number of different types of the globular proteins are present inside of that. Here, next, what it is means the structure. You know, next. The inside the ssRNA is there now. This ssRNA means what it is genetic material. This is actually the genetic material. Here, the genetic material mainly the ssRNA that is synthesized. First, this ssRNA enter inside of the human that synthesize the one single strand. Here inside of the human, they are synthesized single strand DNA complementary to the viral DNA. Means the immediately the virus is enter in our body. Our body mainly in our body the DNA strand is synthesized. Here ssRNA synthesizes the DNA strand complementary to this virus RNA. Here complementary to this the one DNA is formed inside of the human body. The using of which enzyme? Reverse transcriptase enzyme. With the help of the reverse transcriptase enzyme, ssRNA, see here, ssRNA synthesize the first strand. One DNA strand is formed inside of the human. Here, that is this one is complement to the viral DNA. Complement to viral DNA. It is the complement to the viral DNA. That first strand is synthesized. Here, with the help of which one? Reverse transcriptase enzyme. Reverse transcriptase enzyme. With the help of the reverse transcriptase enzyme, ssRNA synthesize the single strand viral DNA, the complementary to the viral DNA is formed. Next, the enzyme, with that enzyme help only, with the help of that enzyme only, they are see the enzyme also help to the formation of formation of second strand DNA, the complementary to the first one. First one is formed. After that, the second one is formed same in the presence of reverse transcriptase only. First, the virus enter in our body. The ssRNA synthesized in the presence of reverse transcriptase enzyme. One DNA strand is formed. After that, for the single strand complementary, the second strand is also formed inside of the human. Here, this is in the presence of the same enzyme. Here, reverse transcriptase enzyme. Then the double strand DNA, viral DNA is formed. This is first it is single strand, it becomes as a double strand. Here, this is called as a double strand. Double strand. Viral DNA, this one is viral DNA. Double strand viral DNA is formed. Here, this see this one, the viral DNA incorporate the host and in the presence of first the double DNA strand is formed. That viral DNA, this is the complement to here the incorporate means they mixed with the host DNA, means human DNA. They are mixed with the human DNA. Here, human DNA. This attached with the first ssRNA forms the single strand of the DNA complement to the viral RNA, and second next with that enzyme only the second strand is also formed. Then that we call as a viral DNA. This viral DNA is also called as the provirus. Here, this is incorporate with the human DNA. It is mixed with the human DNA and form it as a provirus. See this one, double strand viral DNA is formed. The viral DNA is incorporate, means mixed with the host DNA by the enzyme integrase enzyme. Before that, reverse transcriptase enzyme. Here with the help of the integrase enzyme, viral DNA mixed with the host DNA, human DNA. Clear? That we call as incorporate DNA is called as a provirus. Clear? That now we call it as a provirus. This provirus transcript the RNA that can be translated to the viral protein. This DNA is there, this provirus is there, this transcript RNA, the viral RNA is there, this transcript the RNA that is translated to the viral protein. They are formed as the RNA is formed as a viral proteins. Or the particles. These viral particles are gathering, assembled and form it as a HIV virus. Like this, the infected human cells act as a HIV generating factories. Here, there is a TH cells are destroyed. Here, they are infected immediately. The T helper cells are infected and destroyed. Here, this is one more time. I'll explain clearly. Here, the inside of the HIV ssRNA is there. This ssRNA, in the presence of reverse transcriptase enzyme, they are first form it as the one strand of DNA that is complemented to the this RNA. Here, first strand is formed. After that enzyme, reverse transcriptase enzyme only, the second strand is also formed. That is called mixed with the, means incorporate with the DNA, human DNA, and form it as the provirus. Here, with the viral DNA, this is called as both of them are mixed and form it as a provirus. This provirus, they translate the RNA into the viral proteins. Here, they are translate the RNA into the viral proteins. These viral proteins all are gathering and form it as a HIV. Here, this one. Here, the two enzymes are there. One is called as a reverse transcriptase enzyme. It is helpful for the formation of the two strands of DNA inside of the host. They first form the single strand DNA. After that, double strand DNA is formed. That the double DNA mixed with the human DNA and form it as a provirus. Here, what is the use of this provirus? Provirus translate the RNA into the RNA and form it as a viral proteins. These viral proteins are gathering and form it as a HIV. Like this, the infected human cells here inside of the cells in the human, the HIV is every time they are forming. That's why it is called as a HIV generating factories. Here, the T helper cells also damaged, infected, and they also get damaged. Here, next, the time between the first infection and appearance of the symptoms. It is from the few months to 10 years. Means first they are infected now. For the appearance of the symptoms, it may be some months to few months to 10 years. It will be there. Clear? Next is what are the symptoms? Symptoms means all of you know this one. The experience fever, bout of fever, diarrhea, and the losing of the weight is occurred. Clear? Next, due to the loss of T helper cells are damaged now, the bacteria, virus. See, due to the loss of T helper cells, the bacteria and virus which are not deadly invade to the tissues and cause the distress. Means the T helper cells are not perfectly they are control the virus and the bacteria. That's why all types of them, the patients is said to be the AIDS patient. Means all the diseases they can occur. That's why it is called as it causes the AIDS. Here, this one is about the HIV and the AIDS. Next, we are going to the next lesson. Genetics. Next, what means the in the reproductive system and the reproductive health, no SAQs are there. That's why I'm taking the next lesson, next unit, that is the 6th lesson, genetics. Next chapter, genetics. The genetics, 4 to 5 SAQs are there, what we will see. Actually, sexual transmitted disease is also important, but according to the weightage, they are not given. That's why I didn't take. Which it's not there, I think. Okay, it's not here in the PDF. It's not here. That's why I take the genetics here. In the genetics mainly, which answers are important means the first one is the human, the sex determination in human. First, the sex determination in human. Here what it is means, here sex determination in human means already we discussed about the the chromosomal theory of sex determination. In that only same it is there. Here in the human, XY type of XY method of sex determination is occur. Here XY method of sex determination is occur. What it is means, see this one, here the males are the females are the homozygous. Here these are the females. Females are having the 2 X chromosomes. That's why we call as a homozygous. The males are the heterozygous. Here they are having the heterozygous. They are having the different types of chromosomes. X and Y chromosomes are there. That's why it is called as a heterozygous type of sex determination is occur. Next, what it is means here, the XY type of determination is present. See, I'll write it here. AA XX cross with AA XY. Here, this one is the female. This one is the male. Here, the males only determine the sex. What are the gametes will occur here? All the ovaries all the ovas are same, but the sperms are different. Here, the half of the sperms are having the AX chromosome, half of the sperms are having the AY chromosome. Means here the sex determination is mainly in the human by the male only. Male determine the sex. Here, see this one. Here you gather, you combine it this one. Here the AA XX. Then AAXX is formed. This it is the female. Means the ovas all are same, but the ova is attached with the fused with the AX chromosome sperm. The female will be produced. Here, if it is fused with the AY chromosome, AY sperm, then they are form it as a male. Here is formed as a male. This is about the sex determination in the next. How many chromosomes are present in the both of them are having the 23 pairs of chromosomes. Human is having the 23 pairs of chromosomes are present. In this 23 pairs of chromosomes, 22 are the autosomes. 22 are the autosomes. One is the allosomes, are also called as a sex chromosomes. Allosomes. Here, see this one. This is about the sex determination in the human. Here, genetics. This one is about the determination. Here. Next one is the erythroblastosis fetalis. Next one is the in the genetics. Next question is erythroblastosis fetalis. Here, the erythroblast in the fetus, the erythroblasts are destroyed. That we call as the erythroblastosis fetalis. What it is means it is the, for example, the children is having the Rh positive, or the children is having the Rh positive. Child is having the Rh positive, and the his father is having the Rh positive blood group, and the mother is having the Rh negative blood group. Means what is the Rh negative group? Means the already we discuss about the blood groups. Here B, O blood groups we discuss. Additionally, one more antigen is present that we call as a D antigen, or also called as a rhesus antigen. Here it is find out in the rhesus monkey. That's why it is called as a rhesus antigen. How it is means this is also present on the RBC only. Here they are present on the RBC. D antigen are also called as a Rh antigen. Here, for example, one person is having the A positive group is there. They are having the RBC is having the A antigen is present as well as Rh antigen is present. Here, that is called as a positive blood group. Here, like this only. Here, they are having the some are having the Rh positive antigen is there, but some are not having this type of antigen. The Rh antigen is not present in all the persons. How it is means some persons are having the Rh positive on the RBC, some are not having the Rh positive. Which one is having the Rh positive antigen that we call as a Rh positive? Which one is not having the Rh antigen that is considered as a Rh negative? Here, like this, see, observe here, the children, here, the children is having the which group? Rh positive group is having his father is having the Rh positive, mother is having the Rh negative. This time what will happen means while the delivery time here, you observe here between the mother and the children, the blood change will be there. Means mother is having the Rh negative, the child is having the Rh positive. That's why, for example, inside of that the fetus is there. For example, this is a uterus. Inside of that, the children is having the Rh positive. The surrounding this mother is having the Rh negative group. Here, mother is having the Rh negative group. That time what will happen means both of them are the different. Means the fetus is having the antigen, mother blood doesn't have the antigen. That time normally antigens reversely antibodies are produced in our body, but Rh antibodies are not produced before only when it is sensitized. Means for example, Rh positive blood is mixed with the mother blood. Means through the delivery time, through the placenta, they are delivery time, both mother and the fetus blood is mixed. That time mother get it sensitized. Here, mother gets sensitized. Mother gets sensitized. At that time, what will have sensitized means they can try to prepare the RH, the antibodies. Here they prepare the RH antibodies for attacking purpose. The antibodies are produced by the mother. Here, this one is formed. In this, what will happen? Second child, for example, it is a first child. Normally the mother is not able to produce the antibodies. That's why first delivery is a normal. The second time, the fetus is also the fetus is getting the RH positive blood group only. That time, what will happen means already the antibodies are produced inside of the blood of the mother, and that in the second time. This one is the first time. That is the normal delivery is occur. The second time, second fetus, second children time, he is also getting the RH positive. What will happen means mother already antibodies or mother blood is having the antibodies. They enter inside of the circulation of the fetus and they destroy the erythrocytes. Means the erythrocytes are before the immature cells are there. They are called as a erythroblasts. Erythroblasts. These erythroblasts are undergoes to destroy. That we call as a erythroblastosis. Here, erythroblastosis means they destroy the erythrocytes. Means they are before that actually erythroblasts are the immature cells of the RBC. That directly they are released. Means they are not undergoes the mature. Directly they are released. That time the RBC will be destroyed. That we call as a erythroblastosis. That is also called as a hemolytic disorder in newborn babies. See this one, hemolytic disorder in newborn babies. Here, this is about the erythroblastosis fetalis. With the notes, I will explain one more time. Here it is in this it's not there. That's why I am taking. Next, I'll take the no PDF. With the PDF, I will explain tomorrow. Now I am completing time is there now. That's why first I am completing the evolution first. Clear? I'll take the next lesson first. After that, the genetics I will take. Without PDF, you can't understand. That's why I'll take the next lesson. See this one. All of you know this one. This is the homologous and the analog evolution. I'm taking in the genetics or the genetics more SAQs are there. I will take in the next class. The why means no, it is PDF is not available. That's why we'll take tomorrow's class. Now first I'm taking the evolution. From the evolution also, one SAQ is compulsory. Sometimes it gives 2 SAQs also. That's why listen clearly. Here it is, see this one. Homologous means same. The organs which have the similar structure and origin. Similar they are, and the origin also same. Similar origin and similar structure is there, but not necessarily the same in the function is called as the homologous. Means the structure and origin is same. Both of them are formed from the same and the structure also same, but the function is different. Here the organs which are different origin, but they are having the same function that we call as a homologous. For example, insects are having the wings and the birds also having the wings. Here, the structurally they are different. The insect, the wings are having the nervures The bird wings are having the bones provided with the feathers and bones. The structurally they are different, but the function is same. That we call as a analogous. Here, for example, we are having the hands. Human is having the hands. The others are having the limbs are there. Next, the whale is having also the pygidium is there. Here, like this, but they are structurally the origin is same, but function is different. Some are for the walking, some are for the climbing, some are for the swimming, like that. That is means here the what it is. Here the structure is same. Structure homologous means structure is same. Structure and origin is same, but function is different. Function is different. Here in this second situation, opposite. Structure is different. Function is same. Write it like that also, like this also. Here, structure is the same and the function is different. That type of organs is called as a homologous organs. Structurally, they are different and function is same. That we call as the analogous organs. Here, what it is, such organs show the adaptive radiation or divergent evolution. Here, they are formed as a divergent evolution, and these are the convergent evolution. Here, they are structurally different. They are formed the convergent evolution. These are formed as a adaptive radiation. Here, example, they gave here. See this one. The four limbs of vertebrates. Here, four limbs of the vertebrates. Any vertebrate you take, the fishes, amphibians, reptiles, anyone. The four limbs of the vertebrates. The flippers of the whale flippers. The hand of man and wings of bat. Here, the vertebrate four limbs you can take. The flippers is present in the whale. In the whale, four limbs are modified as a flippers. In the man, they are formed as a hand. And the inside of the bat wings, like this, they are origin structure is same, but function is different. Here, this is about the example for the homologous organs. Next, here already I said analogous organs means the wings of butterfly and the wings of the birds. Means the wings of the butterfly, they are having structurally the inside of the wings nervures are present. In the bird wings are having the bones are present. That's why there's a different that is called as a analogous organs. Here, next, all these organs have the same arrangement of the bones. Bones means here what it is: femur, radius, ulna, tarsals, metatarsals, like that, phalanges, same structure is present here. All these organs have the same arrangement of the bones, but their functions vary their mode of life. Here, the structurally they are same, but functionally they are different. Here, when the animals live the same habitat and they load the similar mode of life, they have the same body forms. Means what it is, they are same which one, the same environment they are living. Here, same habitat they are living. For the aviation means for the inside of the air, for the flying purpose, some organisms are wings are present. In the birds also wings are present. Structurally they are different, but in that habitat, for that purpose, their work is same, function is same. That's why they are modified. That is called as a analogous organs. Here, this one is important. The simply homo means same in the structure. Function is different. That we call as homologous. Analogous means structure is different, but same habitat they are living. That's why function is same. That we call as the analogous organs. Clear example: four limbs of the vertebrate here, the wings of the butterfly and wings of the bird. Here, next, in this all the organs are same arrangement, but functions are different. Here, the animals are living in the same habitat, the similar mode of life they are having. That's why functionally same. This is about the homologous and the analogous organs. Here, next. Next one is the theory of mutations. Next one is theory of mutations, or also called as a mutational theory. Yeah. See this one. The what about this mutations means the change, the variations and the mutations, sudden changes are occur. The genetically sudden variations are occur that we call as the mutations. Here variations means they are permanent, but this one is the mutations in that particular generation only they are occur that we call as a mutation. See this one. These are the sudden. See this one, mutation. These are sudden random inheritable changes occur in the organism. Suddenly the changes are occur that we call as a mutations. The mutational theory was proposed by the who? Hugo de Vries. Here, Hugo de Vries. Here, Hugo de Vries proposed this mutational theory. Next, he taking for his experiment, he take the plant that is called as a Oinothera lamarckiana plant. That is also called as a commonly evening primrose. Here, they are having the different forms are there. Here, he found the 4 different forms of Oinothera lamarckiana. In this, some are having the small style form is present. They are having the small styles are having, and some are having the smooth leaves are having, and some are bigger plants that is called the gained form. Some are the smaller plants that we call as dwarf. Here, Oinothera lamarckiana is the only one plant. In that plant, some changes are occurred. With that changes, they are formed as a 4 forms. 4 different forms are formed. Here. See here, first one is called as a Oinothera. O means here the Oinothera brevis stylis. In the brevis stylis, small style form, they are the Oinothera brevis stylis. In that plant, the small style is present here. Next the Oinothera livifolia. In this, the smooth leaves are present here. Next is called as a gigas. Oinothera gigas means in the name of gigas means gained form, bigger plant is present here. Next is the Oinothera nanella. In this, dwarf form is present. Like this, the all are same only. This all belongs to the Oinothera only. Here, the species will different means some mutations are occur. That's why the changes are occur that we call as a mutations. Here, see this one. All the 4 forms are mutant forms only. Here, from they are mutated, undergoes a change and form it like this. That's why we call it the mutant forms. Here, Darwin called the mutations as a sports of nature. Means they are the sports means they are the competition that is called as a. He said that it is the sports of the nature. Next, the Bateson, who Darwin is called it as a mutations sports of nature. The Bateson called them discontinuous variations. Variations are discontinuous. Variations actually they are permanent, but here the discontinuous in the some generations that we call as a. That's why Bateson is called it as a discontinuous variations. Here how it is, Darwin, the mutations are called it as a sports of nature, and the Bateson called it as a discontinuous variations. Here, next what are the main characters of the mutational theory means mutations occur from time to time in the natural breeding population. Time to time they will occur in the natural, not artificial, natural breeding population. Time to time they will be occur. Next, they are already, Bateson said, they are discontinuous and not accumulated over the generation. Here, they are every time, they are discontinuous, they are not continue, they are not permanent. They are the discontinuous and not accumulated over all the generations, they will be not occur. Next, they are full-fledged and there is no intermediate forms. Here, they are full-fledged, there between them, no intermediate forms are there, they are the full-fledged. Here, next, they are subjected to the natural selection. Nature will select automatically the change will, depending upon the environment, depending upon the nature, the changes are there. That's why it is the subjected to the natural selection. See this one more time. We'll explain. Mutations means the sudden change, temporary, that we call as the mutations. Here, these are the sudden random inheritance changes occur in the organisms. Here, that is who proposed this theory. The mutational theory is proposed by the Hugo de Vries. Here, next, which plant he taken for the experiment? The Oinothera lamarckiana plant. That plant, a common name is called as a evening primrose. Here, it is having the, after the mutations, it is having the 4 forms are there. They are the Oinothera brevistylis, Oinothera levifolia, Oinothera gigas, Oinothera nanella. Means they are 4 are having the different structures, different forms are there. That's why we call it these forms. You write it all these forms also example. Next, all the 4 forms are the mutant forms. What the Darwin said, mutations are the sports of nature. Next, Bateson, he said that these are the discontinuous variations. Here, next, what are the features? They are occur time to time in the natural population. Here, they are discontinuous and non-accumulated in the generations. They are full-fledged and they are subjected to natural selection. This one is about the mutational theory. Next, a very, very important question. This one is, industrial melanism. Here, Darwin theory, natural selection, that is how the industrial melanism. Here, under the Darwin theory of natural selection, in this they are called as a industrial melanism, or as a means the Darwin proved the natural selection with the industrial melanism of the organisms. Clear? What it is means the Darwin's theory of natural selection. See this one, Darwin theory of natural selection explain how the evolution might be occur in the nature. For the explaining of the natural selection, he taken the industrial melanism. Here, what it is means, that is ecological example for the natural selection is industrial melanism. He taken the classical example for the explaining of the natural selection, the exhibited on the peppered moth Bison vetilaria. Here, the name is what it is, pepper moth. Pepper moth. What is the scientific name? See this one, Bison vetilaria. Bison vetilaria. Pepper moth, so taken as a for example, and that scientific name is called as a Bison vetilaria. And these moths were available in 2 colors. Here, it is available. How many colors? The 2 colors it is available. Some are black in color, some are gray in color. Here, gray in color. 2 forms are there. Here, these moths were available 2 colors, gray and black. Here, gray moths were abundant before the Industrial Revolution all over the England. In England, before the industrialization, the gray moths are more. Why means no pollution is there. That's why no soot is there. That's why gray moths are more abundant before the Industrial Revolution in England. Here, but after the establishment of the industries in England, black moths are first before the industrialization. The gray moths are more. Why means the tree barks are there, na stem. They are like a almost gray color only. They are settled on the bark. They are not visible to the predators. That's why they are more before that. Before, After the industrialization, the soot come, the tree barks are becomes as a black color. The black marks are settled on the barks. The black marks are not visible to the predators. That's why the black marks are more. Means before the before the establishment of the industries, the gray color is more. After the establishment of the industries, black color are more. Clear this one. See this one. After establishment of industries, England, black color marks were more and gray color marks were less. This is due to the pollution from the industries to form the soot, turn the box into the color black color. Here, so prey means predators. So prey marks were easily identified and were more produced, more predated by bird. Means the gray color is easily they can get. That's why gray color marks are reduced after the industrial melanism. Here, next, thus the gray marks decreased in the number, black marks are increased in the population. Here, this natural selection favor to the melanic marks, means black marks, and to reproduce more successfully. Here, natural selection is a natural selection of the darker forms responds the industrial pollution known as a industrial melanism. Here, this one depending upon the nature, the changes will occur that we call it as a natural selection. The natural selection explained by the it is under the Darwin's theory, and he explained that the nature will select the organisms. How it is means he take the good example means classical example he taken for the explaining of the industrial melanism, and the pepper moth he taken as example. Pepper moth, the scientific name of that is called as a Biston Betillaria. It is having the two colors are available. They are the black color and gray color. The before the industrialization in England, the gray color is more. Means in that time no soot is coming, and that's why tree barks are gray in color. That's why predators are not identifying the gray color. That's why gray color are more. The black color is less in that time. After the industry's establishment, the gray color will be less. Means why means the soot will come through the pollution, and that time the barks are becomes as a black color, and the gray color marks are easily visible. That's why they are decreased. The black color will be increased. Here, this one is about the Darwin's natural selection. Here, this is today we discuss about the 3 questions in the evolution. The one is the all the 3 are very, very important. What first one? Homologous and analogous organs. Homologous and analogous organs. Second is the mutational theory. Third one is the industrial melanism. Darwin's natural selection, industrial melanism. Here, this one is about the evolution. Next, more are there, more 2-3 questions are there. That we will discuss in the next class. This see this one. What is the homologous analysis? Simply, the homologous means structurally same and functionally different. That organs are called as the homologous organs. The example is the forelimbs of the vertebrates. Simply. Next, analogous organs means they are structurally different, functionally same. Example of the means the wings of bird and the wings of insects. Here, that much is enough. The one is called as a convergent evolution, and the second one is called a divergent evolution. Next is the mutational theory. Here, some changes are occur in the organism, sudden changes are occur that we call as the mutational theory. That mutational theory explained by the Hugo de Vries with the evening primrose and Oinothera lamarckiana. In that, he find out the three forms. The names are different. The names are important. You learn it that. And the next one is called as the industrial melanism. This is under the natural selection of the Darwin. And he explained, he taken the example, the pepper moth are also called as a distant Batuleria. In this, two moths are there. The after the industrialization, before industrialization, which colors are more? He explained before the industrialization, gray moths are more, barks are gray color. That's why the predators are not identified. And after that, after the industrialization, black color is more. Here, this one is about the these three questions. Next, the remaining two, three questions are there. SQs are important are there. I will take tomorrow's class, and as well as the genetics I didn't explain. Genetics only. The one question I explained. I think one second question also clearly not explained. Tomorrow, try to explain that one. Yeah. Okay. Tomorrow we continue with the evolution. Yeah.